Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this section to discuss NCERT Class 12, the chapter is Biotechnology Principles and Process. Part 6, let's discuss process of recombinant DNA technology. Process of recombinant DNA technology. Recombinant DNA technology involves the several steps, the specific sequence such as isolation of DNA, fragmentation of DNA by restriction endonucleases, isolation of desired DNA fragments, and ligation of the DNA fragments into vectors, and transferring the recombinant DNA into host cell, and culturing the host cell in a medium, the large scale, the extracting desirable products. So they followed as in these all steps. First one is what? Isolation of DNA. After that, fragmentation of DNA, the restriction to nucleus. Third one is what? Isolation of desired DNA fragments. Ligation DNA, the fragmentation to as in a vectors. And next, transferring of the recombinant DNA to as in a host cell and culturing as a host cell in medium large scales, extracting from as in a disable product. So these are the complete steps involved in the process of recombinant DNA technology. Okay, let us there examine each of these steps some details. So let us discuss clearly in a step by step what they going on in this total recombinant DNA technology process. Okay, in that first one is what? Isolation of genetic material, that is what? DNA. Isolation of DNA. Let's see this diagrams also they given here clearly. Here they're completely their DNA is separated, can be using as any removal of by as any spooling process. Okay, let's see. First, isolation of the genetic material DNA. The recall the nucleotide, the genetic material in all organisms without expression. So already we know that all organisms they contain as a genetic material that is in a DNA, and some RNA also nucleic acid simply. Okay, here the majority of organisms this is in a deoxyribonucleic acid or a DNA. Mostly has in what? They contain as in it the DNA is get present. Let us. In order to cut the DNA with as an restriction enzymes, I need that it need to be as in a pure form, the free from other macromolecules. So when take as in this DNA is completely pure? No, they completely has in the number of bacteria and fats and other bacterial wall layers. Everything is present. So how do they get completely purified? Let's see as here. Such as since the DNA enclosed with the in a membrane, we have to break that in a cell wall, open the release the DNA with other macromolecules such as any RNA, proteins, polysaccharides, and lipids like. So the mostly the DNA contain as what? The DNA mostly they here contain as the number of the walls they are completely they are made up of. The cell wall is made up of number of the cells, protease and like they complete the different types of cell walls they are present on the DNA. So how do they get break? Let's see here. See, this can be achieved the treating the bacterial cell, the plant cell or animal tissues, the enzymes such as in a lysosome, bacteria, cellulase, plant cell, chitinase in fungus. So when we are using as in this enzyme, when enzyme to get add here, what happen? If in case the bacterial cells, the cell wall is present, the lysosome they complete to get digest that layers. If in case the plant cells, the cell wall is present, the cellulose to get digested to as any that's layers. Next, the fungus is present, the chitinase to get digested as in their cell wall layers. So once their cell walls they are completely absent, what happened? They are finally be getting as in a completely purified DNA will be get. Okay. Now their genes are located on a long molecule of the DNA, the interview with as in a proteins such as in a host to that is histones. So they're finally when they are completely clear what happened, then finally you get as in a they are the long DNA fragments or to get obtained. Okay. They contain as what number of histone proteins also present. Now, after the RNA can be removed by the treatment with as in a ribonuclease, where the proteins can be removed by as in a treatment with as in a protease. So RNA, the single stranded the ribonucleic acid, if the single strand is present, so we get add with as in a protease enzyme. We get add with as what? The protease enzyme is add. What happened? The protease enzyme to get digested to as in that single layers. Okay, the other molecules can be removed, they are appropriated the treatment, that is purified the DNA. They are ultimately the participate the out after addition of an ethyl ethanol, they can be seen in collection of in a fine the thread into as a suspension. So after in this total medium, the DNA fragments are comes here to get add with as what? They get add with as in a chilled ethanol. So once chilled ethanol add, what happen? The enter the purified the DNA fragments we are notice. Simply here the purified the DNA fragments we are completely gets. Okay, this is the in completely first one, the isolation of the DNA. So finally we getting as what? Completely purified DNA are getting. Next one, that is cutting of DNA at specific locations. Next one is what? Cutting of DNA at specific locations. The restriction enzyme to digestion they are performed as an incubating the purified DNA. They include the molecule with as an restriction enzymes. So after when you get as an that complete the DNA, when you get as an the complete the purified DNA. Now here that specific enzymes we get used for as in the cut the DNA. So this DNA to cut with help of as in specific enzymes. Let's see here agarose gel electrophoresis employed the check their process of any the restriction enzyme digestion. So we are using, if in case when cut the DNA, we get as in what the small DNAs comes and medium and the small DNA fragments also get, right? So when you are using a gel electrophoresis method, previous we discussed, right? So the gel electrophoresis to automatically to get separate as in that DNA fragments. Okay, next the DNA is negatively charged molecule, hence moves towards in a positive completely in a anode region. Yes, right? Get reached as in a positively anode region. This process repeated with as in the vector DNA also. So here lnDNA and vector DNA same we do as in the same process, means restriction enzymes, and then when we are using as restriction enzymes to cut the DNAs, and the DNAs also to get separate with help of in a gel electrophoresis. Next, the joining the DNA involves the several process after having the cut the source of the DNA as well as in a vector DNA, the separating the restriction enzymes. So, like next, the cut out the DNA insert into as a source of DNA vector with as an space of the mixed and ligase add to result as any participation of recombinant DNA. So finally, we this cut the DNAs and finally to get add with as in with the help of in a ligase enzyme to develop as in the new DNA. That is DNA is called as what? Recombinant DNA. That is in a or DNA. Recombinant DNA is to get developed. Okay, this is in a cutting of DNA at a specific location. Next, third one is what? Amplification of gene insert into as any using PCR. Amplification of gene of interest using PCR. Here PCR stands for as what? Polymerized chain reaction. Polymerized chain reaction. In this reaction, there are multiple copies of genes or DNA insert into synthesis of as an A. They are in vitro to using as an A to using in the set of the primes in a small chemical synthesized as in a oligonucleotide. There are in a complementary with region of DNA and enzymes of in DNA polymerase. So in this we are using as in a complete DNA polymerase enzyme. So mostly in this PCR they get followed as in three steps in this process. The first one is in a denaturation, means to give as in the more temperature around as in a 90 to 95 degree the temperature. When you give, what happen? This is a denaturation process. What happened? This total two strands will be to get completely to their breakdown. Then what happened after to get annealing process? Annealing process means to get give as any completely in normal conditions. To give as in normal conditions, then what happened? It is to get forms as in their small their primes are formed. Small primes are formed. And last one is what? Extension. Third one is what? Prime extension. In this prime extension, what happened? In this prime to get complete to get grow here, and is completely growing here. So then what happened to get completely to get produced as an A? There two DNA fragments as produced with help of enzyme is what? This is a Taq polymerase enzyme. That is what? Taq polymerase, Thermus aquaticus polymerase enzyme is we are get using. Taq polymerase enzyme. So after in this denaturation process, when you do as in there 30 cycles, we get as an around as an amplified into as an 1 billion times. They get 1 billion times to get produced as a number of copies. Okay, now this is in a completely the process of in a PCR. Let us see the enzyme extends the primes using as a nucleotides provide in the reaction and the genomic DNA template. So in this total nucleotide, when we are doing process with the help of what? Genomic DNA template. If, if the process of the replication, the DNA is repeated many times, so the number of times means around to be using as 30 cycles, am right? So repeating times, the segment of the DNA can be there amplified approximately the billion times. They get what? The billion times the multiple copies we are getting. 1 billion copies are made such the repeated amplification they are achieved by using as in a their thermostable DNA polymerase isolated from as in bacterium Thermus aquaticus. So that polymerase, so we are using as in thermostable bacteria. That is what that is Thermus aquaticus. It is get using that is to get help for as in what that is complete to get help for as in to get formation of the multiple copies, which is remain there active during the in a high temperature, including the denaturation of the in a double stranded DNA. So high temperature, what happened when we are using as a 90 to 95 degrees centigrades here to complete the one DNA fragments to get divided into to form as a two strands. Am I right? That is, and here amplified fragments to dessert can be now to get used the ligated with vector for the cloning process. So that finally what happened, the number of how many the copies we require there they get producing with help of in a cloning process. Okay, this is next one. This is a complete amplification process. Let's we discuss in the insertion of recombinant DNA into as a host cell or organism. Here the insertion of recombinant DNA or DNA. Let's there is a several methods the introducing the ligated DNA into as an recipient cell. So here how do they introduce that? Before also we discussed what is that. It is a process of in a bialistic method, means that is gene gun method and micro injection method we discussed. Am right? Same like this one also say. Here the recipient cells after making them they're competent or to receive to take up the DNA present in the surroundings. So surrounding the complete DNA we are identified. So the recombinant DNA to bearing the gene the resistant to the antibiotics. So ampicillin genes we are present. Example they given as an ampicillin genes as present. The total genes we are complete their uptake and it is transferred into as an E. coli cells. It is transferred into where? Into as an E. coli cell. The host cell become as an transferred into as an ampicillin resistant. So that cells also to get converted as an antibiotic resistant will be they get develops. Next, if if we they spread the in a transformed cells in a agar plate contain medium ampicillin, only transformed cells will be grow, untransformed cells will be what happened to get die. Only ampicillin will be good grow, but another the tetracycline does not there grow. Since due to the ampicillin resistant genes is one able to select and transformed cells in the presence of ampicillin. This ampicillin resistant genes they can case is called as in selectable markers. So this ampicillin genes in this case we call as what? It act as in selectable markers. Selectable markers. Next, remaining we discuss in next class. Thank you for watching. Please share, subscribe and like. Thank you.